Gene expression and cloning will be the main topics of this chapter in this review video. So in the very first part of this chapter, they talk about cells and how every cell in your body has exactly the same instructions, exactly the same DNA instructions. So when we look at the different cell types, and we'll move on to this next slide here, we look at muscle cells, pancreas cells, blood cells. Why do they look so different? Well, the major reason that they look so different, and more importantly, they really function so differently, is because some of the genes, parts of the DNA, for instance in the muscle cell, are active, and you're actively making proteins through transcription and translation from certain set of genes or certain part of the DNA in those cell types. And in, for instance, pancreas cells, you're using another part or a different combination of those genes. So it would be similar to taking a dictionary. So every cell has a dictionary, okay? The same dictionary. However, each cell writes its own book from the words in that dictionary. Does that make sense? I sure hope it does. That's, I think, one of the best analogies I can think of to describing this, the reason for this difference that we see, being as though every cell has the same set of genes, has the same DNA. All right, so we're going to move on to cloning now, this idea of taking an organism, using its genetic material, using the genetic material of an organism that already exists, and either making a genetically identical organism, or, which is more often the case, especially in research situations, using those cells from the embryo to make cells of a certain type, which is therapeutic cloning. So when we look at cloning, there are a lot of benefits, but especially in reproductive cloning, there are many ethical issues that are involved, and we'll talk a little bit more about those ethical issues in just a moment here. But with animal cloning, both reproductive and therapeutic cloning start the same way, and you're really going to want to understand the steps involved here. I believe there are 5 steps involved in this process. We start here with removing the nucleus from an egg cell. So you're taking out that haploid cell's nucleus. As you can see here in step 1, that little purpley circle is the nucleus. And then the key step here is you take a donor cell from whatever organism you want to clone. Either clone it completely into a full organism or just clone its cells for therapeutic uses, and you inject that into the egg cell, which is shown here in step 2. And then you allow that egg to develop, to grow by mitosis, divide, when put into a particular medium of nutrients and molecules that activate the division of these cells, and you produce an early embryo. So again, this is for both therapeutic and reproductive cloning. Now with reproductive cloning, what you would do is you would take that early embryo that you made in step 3, go on to step 4, and you would place that embryo into the uterus of a surrogate mother. Okay? And then of course that embryo would develop into the clone, and you would have a clone organism born. That is reproductive cloning. Now again, this is really not the goal of most scientists. Most scientists are after this therapeutic cloning modality. So what you want to do here, what they do with therapeutic cloning for the most part, there are some advances that are going on with this, so that embryos may not in the future be used for this process, because there's a lot of controversy here, because you take that early embryo and you break it apart so that it can no longer become a full organism, and you take the cells that would have become the organism, which have not differentiated just yet, they haven't differentiated in any particular part of the organism, and you put them into a medium that directs their development into a certain cell type. So for instance, if you're trying to reproduce cells for spinal cord injury individuals or Parkinson's disease, recreating those types of cells in an effort to come up with therapies for these particular individuals, that would be therapeutic cloning. So you're trying to help particular individuals by creating just one type of cell. All right, now reproductive cloning, there are some valuable applications here, but a lot of times, again, we're really not trying to reproduce a full organism. What we're really after more is this idea of therapeutic cloning, and there's controversy in this because, again, you're destroying that embryo. And now these cells are so valuable, they're called stem cells, these cells of this early embryo, because they, again, can become any type of cell in the body, any type of cell. And you can direct their growth or their development into a certain cell type that you'd like to study or like to use in a therapy for someone, again, who has a spinal cord injury perhaps, or Parkinson's, or any one of a number of chronic illnesses that really have no cure at this time. Now there is some more recent research into adult stem cells. So as adults we do have stem cells, mainly in the bone marrow, and these cells can be cultured to become certain cell types, blood cells for the most part. And there's other research that scientists have taken, for instance, skin cells of mice, and they have de-differentiated them, which means they were using a certain set or certain words from the dictionary, and the scientists took that cell back to using the full dictionary. So it de-differentiated it, and then it exposed the cells to certain molecules, certain chemical elements that cause that cell to re-differentiate into a certain cell type, and now it's using different words from that dictionary. So it's all about this idea of what parts of the code are being used, and that's what's going to give you a certain cell type. So adult stem cells, again, we do have them in our bone marrow, and there is some research, which is not indicated here in this diagram, that scientists have taken cells that are differentiated, say a nerve cell, and or a skin cell, which is actually used in this experiment, de-differentiated it back to a stem cell-like nature, and then re-differentiated it into a nerve cell or a heart cell. So there's still a lot of research, and it's really in the very beginning stages here, but this is really just an overview to give you an idea of the difference between an embryonic stem cell and then an adult stem cell, and the controversy involved in stem cells and stem cell research. So be sure you watch the video that is on the Isidore website for your viewing pleasure, and if you have any questions on these topics, please don't hesitate to ask.